Hi everyone, my name is Franco and I'm the head of the mass spectrometry facility here at the LMB, and I'm going to try and keep it on my place. So first thing I want to ask you, and if you by any chance have your cell phone with you, this is actually quite useful because this is interactive. You can scan the QR code and hopefully you'll have a question popping in your cell phone. Let me know if you're able to do this or not, because the first question that I want to ask, and just to make sure that everyone is well and awake, is what does mass spectrometry measure? Oh, we have two answers. Well then, and that's correct. We do measure mass to charge ratio. I was kind of hoping that you would go tempted and go press mass, but no, we do measure mass to charge ratio. In fact, if you don't have any charge, we won't be able to see anything inside the mass spec. It needs to have a charge, or many charges are the negative or positive. So it goes inside the mass spec. There we go. So if we measure mass to charge ratio, how can we then determine the mass? Well, if you have a mass spectrometry which has enough resolution, you actually are going to see the isotopic series of your molecule. And because carbon is obviously the most abundant element, and as you know, at least 1% of the carbons have a carbon 13 instead of a carbon 12. And this means that you're actually on the mass spec, what you're going to see, instead of just one peak, which we call the monoisotopic peak, you're going to see a series of isotopes. And the difference, if you're thinking about a singly charged molecule, the difference between the first isotopic peak, which is also called the monoisotopic peak, which has only carbon 12, and the difference of the next isotopic peak, which has one carbon 13, if it's a singly charged molecule, the difference is 1. Well, that makes things easier because then it means that we can determine the charge. Because if we look at the difference, and now we have a doubly charged molecule, the distance between the first one is now reduced to 0.5. And then on a triply charged, quadruply charged, and it goes like this. So it's very easy. If we look at the isotopic distribution, and if you have high enough resolution to do so, and this gets complex and very complicated when you have very large molecules such as intact proteins or even virus capsules, which we can also do that with specific mass spectrometers, then you can see the isotopic resolution, you can determine the charge, and obviously from knowing the mass over charge, with the charge state, we can then calculate the mass. That's a very easy way to do. So many molecules can be identified by their mass, but also many molecules are isobaric, which means they have exactly the same mass, which means that you end up with many solutions to one masses. What mass spectrometry also does is we fragment these molecules, and then we get from the fragmentation pattern, we can build which parts of the molecules are constituted of the molecule. Hopefully one of these parts will be unique to that specific molecule, and we can identify the molecule. In terms of what we can analyze, as I said in the beginning, it needs to be an ion. It could be on the solid state, depending on the ionization source, and we'll speak a little bit about this later on. It can be on a liquid and on the gas phase. And in terms of size, we can measure very small things, such as drugs and metabolites, up to what we could potentially call molecular whales, because they are really, really big. And some mass specs can actually measure the mass of the things as big as virus capsids. So it's extremely big, as you can see. And here you can actually see that the average charge distribution on a molecule like this is around 165 to 110, so it's you need to have a really high resolution, and in some situations you actually need to have a slightly different operating mass spec. Right, so second question, for the ones who just arrived, you can use the QR code in your cell phones if you have. If you don't worry, this is just to make things slightly funner. So now my second question to you is, how many components does a mass spec have? And you can just go wild here. I'm going to go ahead. I'm going to say, probably I'm going to surprise you or not, that the mass spec has 3 main components. It has many more parts, but basically you can characterize a mass spec based on 3 main components: the ionization source, the mass analyzer, and the detector. That was easy. It seems very simple. So now let's dig in a little bit more detail in what is one of each of these components. Ionization source. Well, as the name says, the ionization source is the component of the mass spec that is the driver of the ions on its matrix in which they are inside the mass spec. And here I can show you, this is actually an ionization source where we actually plug our columns in here, and on the front end, you're going to have an emitter, and I'm going to show you how this looks in a minute, in which is going to pulverize your sample inside the mass spec. And there's many different types of ionization sources in mass spectrometry. And I would say that the two ones that were the key drivers into being able to analyze peptides and biological molecules in mass spectrometry were matrix-assisted laser desorption ionization, MALDI, and electrospray ionization, when we have both of these ionization sources downstairs. This is an electrospray ionization source. And it was John Fenn and Tanaka Koichi were the awardees of the Chemistry Nobel Prize in 2002 for both these two soft ionization sources, which is exactly what enabled the current state of proteomics in nowadays. It started everyone was using MALDI ionization back 20 years ago, probably, when we were doing identification of proteins in gel bands. But now the community has kind of used MALDI to do a different type of analysis, such as MALDI imaging. So basically, you cut very thin slices of tissues, and then you could crystallize with a matrix that enables to absorb the energy from an UV laser, and then it absorbs their molecules, and they get analyzed once they hit the mass analyzer. Most of our proteomics downstairs is done by electrospray ionization, and this is how we analyze both peptides and proteins. So how does electrospray ionization work? And you can see here. So in the emitter, imagine that you have a very thin metal capillary in the front. In the emitter, what happens is that we apply high voltage, and this is around 2 to 3 kilovolts. It's quite high, quite dangerous. But what happens is that your analytes are already ionized, because at this stage, if you are working with proteins, they are in pH 2. So basically, when you apply a very high voltage and you have a lot of electrostatic repulsion as well, it forms this cone. And then at the tip of the cone, as you can see, it starts releasing very small droplets. And the electrostatic repulsion of your analytes are going to drive these droplets smaller and smaller until you have individualized ions that then go inside the mass spec. So what's the second component? The mass analyzer. And this is considered to be the heart of the mass spec. And again, there's many mass analyzers available. Another key thing that's interesting is that obviously you need a lot of things. The mass analyzer needs to be bridged between the entry of the mass analyzer, or the entry of the mass spec and the mass analyzer. And this is done by ion optics. And basically, it's something that looks like this. This is actually an ion mobility cell, so it's slightly different. It can look like this, or it can look like this, depending on the different mass spec that you might be playing. And what happens is that they have different voltages applied so that they suck in the ions, your ionized molecules, until they get to the mass analyzer. So mass analyzers actually do the real job. They separate and they analyze mass to charge ratios. The mass spectrometries are usually called by the name of the mass analyzers that they have, and there are many of them available. Currently downstairs, we have both Orbitraps and 2 time-of-flights, and eventually, hopefully one day we'll have an asymmetric track looseness, which is one of the latest generation of mass specs. Mass specs can have solo analyzers, so it can be just one analyzer, or they can be hybrids or tribrids, in which they combine the different capabilities of the different mass analyzers. So for instance, they can be a quadruple that looks like this, which is a very good mass filter. So basically in a mass spec, and we have most of our instruments downstairs are hybrids, that they have a quadruple mass analyzer and something else. It could be a time of flight, or it can be an Orbitrap, or it can be an ion trap. So this is how it looks, a quadruple. And how does it work? And I'm just going to briefly explain the Orbitrap because we have many, and I don't have time to explain all mass analyzers. But in Orbitrap, what happens is your ions go inside the mass spec, And the orbitrap is the one that you see the ions are circling around. And the electric field in the ion trap, it's in such a form that the ions are going to have a very stable circular movement around the central spin. And when this happens, we record what's called the transient. And the transient and the frequency of this transient is characteristic of the mass of a charge of that specific ion that's being analyzed. And you can analyze many ions at the same time. It's also a high resolution mass spectrometry because the transient, the mass spec usually only listens to a very short window of the transient, but the transients can be really long. So depending on how long you're listening to the transient, or the mass spec is listening to the transient, we have higher or lower resolution. And this determines the intact mass. There's more things we can do with it. So we can fragment, and that's what I'm going to show you. And for instance, what happens? So if this is a mass spectra, each of these peaks is one of your analytes. In this case, let's think that there are peptides. What it does is that this one that you see over here, so this is the quadrupole. It actually works as a mass filter. And what the mass spec is going to do, or what we are going to tell it to do, is select one peptide or one ion, and then get rid of everything else, and then go and fragment this one. Let's put the animation to run. Go and fragment this, and then send all the fragment ions onto the orbitrap so I know exactly what are the constitutive parts of my peptide. And this goes on for what we call a cycle of acquisition. So it will select which peptides you want to fragment, it will do it until a certain number, and then it will start again, do the MS1 to see what are the peptides that you have in your sample, it will select again, and it will start to fragment again. So the whole thing is called a cycle of acquisition. And in terms of fragmentation methods, there are many available. We usually do use CID, or collision-induced dissociation, or HCD, which is exactly the same thing. It's just a vendor that's slightly different in terms of the terminology that they use. It's also slightly different in the way that the fragmentation is induced, slightly higher energy. So basically what happens is your ions are selected, they drive into what we call IRM or collision cell, and they are accelerated, so they have lots of kinetic energy, and they're going to bump into inert molecules, usually gas molecules, and they're going to fragment. And at that point, we then send them again to the mass analyzer, and we analyze their masses. And if you think, depending on the fragmentation that we are using, usually if it's CID or HCD, it's usually targeted at the, the peptide bond. So if you see the mass differences between one peak and the other, you're going to see that's exactly the mass of one amino acid. And that's how we sequence peptides. So we get the information both on the mass of the peptide, but this is not enough, trust me, because there will be a lot of isobaric peptides in your sample. So you have to fragment and get that sequence information out of it. And the last component, and I'm definitely not going to speak about a lot about this, is just a detector. It's just the bridge that allows you to get a signal. And in this case, it's just an electron multiplier, and it exists in many different machines, not only in mass spectrometers, where you just have an electron that's going to go through the D nodes and it's going to amplify the signal until it can get to the processing computer and be analyzed. So another thing that's not really part of a mass spec, but it's definitely necessary, especially if you're thinking about proteomics, is to have an hyphenation with an HPLC. And the reason why is that we don't have fast enough mass specs that you can just shoot everything inside, fragment everything, and analyze everything in just one go. We need to buy time to the mass specs so that it's able to do its cycles. So what we do is we inject our sample in an HPLC, usually in reverse phase if we are working with peptides, and it will separate the peptides like this. You get a chromatogram, and in the meantime, at the same time that the peptides are going inside the mass spec, it's going to do its cycles. So the faster the mass spec, the shorter this chromatography can be, the slower the mass spec, the longer this chromatography needs to be. The longer, it means also that your signal goes down because the peaks get broader. Higher peaks are okay, but lower peaks, they will just go missing. So the faster the mass spec, the better your chromatography, the better your results. So just to recap, mass spectrometry measures mass to charge. Molecules, biomolecules in our case, can be identified by their intact mass, but also by the fragmentation products. It's actually the key driver for identification because we have a lot of isobaric components in our samples. Usually if you look at one mass, you have at least 1,000 peptides that could fit into that mass. So it's really important that we fragment to identify. A mass spectrometry has three main components: ionization source, mass analyzer, and detector. And these are usually the ones that characterize the mass spec. It has many more components, but these are the key ones. And it's usually affinated to an hplc because we need to give the mass spec time to analyze our samples. Right, now I'm going to speak a little bit about what we do. And I would say that in our facility, we focus mainly on proteomics and structural proteomics, which are actually very similar to one another. They both work in bottom-up approaches. And bottom-up is a way to say that you have your sample, you digest it in order to have smaller chunk of your proteins into peptides, and you analyze these peptides, either just for a standard quantitative proteomic analysis, or for a structural characterization in which you first cross-linked your sample, or you're labeled with your sample with deuterium in order to see what are the different regions, what are the regions that are flexible or not. Basically, they work in the same way. In terms of sample types that we can analyze, well, it can be something as simple as a protein in a band. You purify your complex, you want to see what's there. What's that weird band that you're not expecting? Is it a truncation of your protein? Is it a contaminant from your expression system? That's something we can help you. We work a lot as well with interacting partners, so doing IP analysis. We usually do standard quantitative proteomics either on cell culture or in complex tissues. Either of them is absolutely fine with us. In terms of tips, I want to give you a couple. So when you're working with gel bands, it's really, really critical that you're working with a very clean recipient where you put your gel in. And this is because it's very prone to contamination. And usually we get a lot of keratin. And you think, oh, keratin, this is coming from my hair. Actually, probably not. If you would see a hair, you probably could remove it quite easily. It's from the dust that's on your workbench, is on the environment around you. So as clean as possible your workbench and the place where you're going to cut your bands, the better. In terms of the interacted proteins, controls. So it's never enough to say that you need to really control your experiment, either, and that's up to you, that's up to your experimental design, either you slightly change your bait in order to have something there that can mimic what are the potentially not non-specific interactors. You need to account for those. Usually, IPs use detergent in their wash steps so that you're washing off these binders that are not specific but are still there and you want to get rid of them. The only thing that we are going to tell you is that's fine. If you can, it's a big if you can, please add 3 more washes without detergent. And the reason why is because detergent suppresses completely ionization, and that's very easy to understand. Negatively charged peptides are going to be positively charged, they're going to interact, and we won't be able to see anything. So if you can, add 3 more washes without detergent, everything the same. But if you cannot, and this is the case, which is very common, I'm working with membrane proteins, I really don't want to remove detergent, otherwise everything will crash. That's fine, as long as you tell us detergent, because we can clean for it. If we don't know it's there, we won't clean for it, and you won't see anything because there will be detergent there. And it's quite difficult to remove. But if we know, that's fine. We'll clean for it. Right. And when you're doing your standard quantitative proteomic analysis, do wash your cells. So you can collect them, wash 3 times in ice cold PBS, and then you can bring us the cell pellets, basically. That's absolutely fine. But please wash them. So now a little break. Question for you, the same scheme. Right, so I'm going to tell you, it depends on the experiment. It depends on your system. It depends on the variability of your samples. If you're working with cell culture, usually the biological variability is quite low. So you'll probably be okay if you're giving me 3 biological replicates. If you're coming to me and saying, look, I've just tried this experiment, I have no idea if it works. Is it okay if I bring you just 1 sample? Yeah, that's absolutely fine, as long as you don't want to do any statistical analysis on it. So what we usually do is, let's have a look first. you can bring me one sample just to see if everything is okay. There's no major contaminants. We actually see something on your sample. And then please, let's do 3 above. It's always better to make sure that we can account for the variability within your sample, because that's actually what's going to dictate the number of biological replicates that you need. If you're working with mouse, that usually carries a lot of biological variability. It's normal. It's a whole organism. So probably need to do n equals 5, sometimes n equals 6. But we'll work with you. So this is a dynamic process. Sometimes it works really well, 3 biological replicates in one go. Other times we need to repeat the experiment, and that's fine. The next one. Okay, so 2 people say 1 microgram, that's too low. That's too low for us. Of course, this depends again, what is your goal? What are you working with? Because if you're telling me, well, but I work with single cells, I want to do single cell proteomic analysis, obviously, we don't have the method set up yet because we don't have a mass spec that's sensitive enough, but obviously that you cannot multiply. But if you're working with an IP sample, potentially you can scale up without any problems. You just have to work a little bit more in order to make sure that your scaling up is not actually ruining your experiments. For standard quantitative proteomic analysis, we usually work between 100 to 300 micrograms. And the reason is mostly driven by what do you want to know. If you're working on the whole large-scale standard proteomic quantitative analysis, maybe 50 micrograms is enough, because what we'll do is we'll multiplex your samples. So in the end, we end up with 1 milligram that we can inject in the mass spec. But if you want to work in characterizing post-translational modification events, such as phosphorylation, then we actually need to drive this number up, because what we need to do is we'll multiplex your samples. We end up with a lot, but then we have to enrich for phosphorylation, for phosphorylated peptides. So this means that you have to start with a large amount in the beginning. And the last one. I got you. No, not yet. We're actually going really fast towards that direction. But remember that in mass spec, we cannot amplify our answers as you can do in Western blot. In Western blot, everything is amplified, not in mass spec. Unfortunately, one peptide is that peptide, and that's going to be your analytes. There's no way we can go above that. However, every single year, there's a brilliant advancement in each of the mass spec companies is driving forward, even more sensitive in a faster mass specs. So we're getting there. But in comparing with the mass specs that we have in the lab, usually Western blots or mass spec is at least 10-fold lower in terms of sensitivity, which means that if you run a Western blot and you have a really low abundant protein that you want, you need to know what's there. You actually have to give us a little bit more sample than what you would load on a Western blot, so we can get there. And I think this is the last of the questions. So through this workflow, I already spoke a little bit about this. So basically, a workflow in almost all of the sample preparation that we have in the lab requires that we do some sort of sample homogenization. This can be done on your side. For instance, if you're doing an IP, you have to do it on your side, but we can do it as well, either by beating, using disruption methods. We can sonicate as well, so that there's many ways of basically increasing the surface area in order to extract as best as possible your proteins from your sample. We can use detergents. That's absolutely no problem as long as we know they're there. We can use chowtropes as well, mass spec compatible detergents that also exist, such as rapid gests from waters. We reduce and we alkylate systems to make sure that we're completely unfolding everything. And the reason why is because then next we are going to digest. So the more unfolded your protein is, the better chances we have to see their peptides. And we usually use trypsin and lysine so that we have arginine and lysine terminated peptides because they are easier to ionize. And then once we do the digestion, off it goes. Sometimes we need to clean up depending on what your sample has, if it has detergents or not. But usually, or we can even do enrichment depending on what your goal is. If you want to analyze phosphopeptides, we have to enrich for those. And then we do the mass spec and the bioinformatic analysis. So in terms of proteomics, usually the thing that we do is relative protein quantification. There's also another mode of quantification, which is the absolute quantification. We usually don't run that, so I'm not going to speak about that. But that just involves having your synthetic peptides that are heavier than your endogenous peptides or the peptides of your protein, and then compare the abundances. So that's absolute. We don't do absolute. Usually we can, but we usually don't. We do relative quantification, which means that what we compare is the abundance of that protein across all your groups. We don't usually compare the protein abundance across your sample. And the reason why is because peptides actually ionize differently. So if we are comparing peptide A from protein A with peptide B from protein B, they actually will have very different intensities. It may not actually correspond to the protein abundance. So we have to be careful always to compare apples to apples, oranges to oranges. Having said that, there are ways to analyze data so that you can actually get some quantitative information across proteins. Right, so relative protein quantification, it can be done either in a labeled way or in a label-free way. And I'm going to explain the both, because that's what we do in the lab. So in a labeled quantification, you can use labels, and they are either metabolic labels, which are introduced really early on into your workflow. It works quite well if you're working with cell culture, because you can actually include stable isotope amino acids in cell culture. Cells are growing, will incorporate these heavy amino acids into their proteins. So you'll have one sample that's going to be heavy, and then another sample that's going to be light. And then what we do is we combine these two before doing anything else. And then we process everything at the same time. And you also have another labeling strategy, which is using tandem mass stacks, TMT. We do quite a lot in the lab as well. It works with any type of sample, basically, whereas stable SILAC for amino acids in cell stable isotopic label amino acids in cell culture only works in cell culture. TMT works with everything. Basically, you collect your samples, you process them, do the digestion, and then you can label the peptides. And at that point, you can combine all your samples and analyze them in a mass spec. Or you don't use any labels at all, and you do what's called a label-free approach. You never combine them. Every single sample is processed individually and goes inside the mass spec individually as well. So this has several advantages and disadvantages. Obviously, the labeled approaches are more expensive. SILAC is not, you cannot use SILAC for everything. TMT is extremely expensive. You can use it almost for everything. LFQ, you're basically using mass spec time, which is also expensive, depending on the mass spec that you have. This is the scheme. It's actually interesting to show. So basically on the SILAC approach, you are controlling for everything. All the variability from your experiment is controlled, because the first step, what you do is you combine your samples. And then you do the digestion, the enrichment, the desalting if you need to, and off it goes into the mass spec. And on the spectra, you'll be able to see the light and the heavy. So on the same spectra, you're actually comparing your samples. It can't get better than that in terms of quantitative accuracy. In TMT, again, TMT is an alternative for everything else that's not in cell culture. There's also another caveat. So there's a limited amount of labels that you can use in SILAC. So you can multiplex 3, 4 maybe. So that's not a lot. There's not a higher degree of multiplexing. You also have the TMT. So chemical labeling, which you label at the peptide level, and it's only after this that you can multiplex your samples. So TMT molecules are composed by an amine reactive group that's going to bind to your peptides. And then it has a balance group and what we call a reporter group. And the addition of the mass from the reporter group and the balance, it's the same. Basically, what changes is the mass of those two. But when it's all intact, they're isobaric, which means that you're going to use isobaric molecules to each of your samples. Each of your samples is going to be labeled with a different isobaric molecule. Well, and then you combine them. Well, if they're isobaric, what's the logic of that? Because that's what we measure. We measure mass. If they're all the same, how can this be informative? Actually, it is very informative, because when you fragment the peptides to get the sequence information, You have also these reporter ions that are coming off from the TMT levels. And the reporter ions carry different heavy molecules, which means that you actually have a flavor of 18 different labels. So you can label 18 different samples, and you can actually do plexes of these 18 to combine all of your samples. In the future, I think some things will change with this method as well to get more higher plexing abilities. So you multiplex your samples. Now, because we have actually a lot of sample, so usually once we multiplex everything, we can have around 1 milligram or even more. So what we do to get more into the depth is we then inject on a preparative HPLC, we fractionate our sample, and then the same sample with everything combined goes inside the mass spec, depending on the mass spec itself, either 12 fractions or 18 fractions. So it can really go down in terms of the depth of the proteome that you're analyzing. Usually we're able to get 8,000 to 9,000. That's almost a complete proteome characterization, but it takes us 3 to 4 days of data acquisition because your sample is going to basically be injected 18 times. And then off you go, you get your report, and you get the identification coming from the first fragmentation. We actually do a third step of fragmentation to have the reporting intensity for the quantification of your samples. In terms of quantification, actually is really good. There's a part of the variability within the sample preparation that's not accounted for. So everything from sample collection until digestion, they have to do it separately. It's only when you label and then multiplex, everything is controlled from that point onwards because everything will be together. Now in label 3, we can acquire this in 2 different ways in the mass spec, what we call a data-dependent acquisition and what we call data-independent acquisition. And this basically also is very dependent on the mass spec that you have. We have a lot of arbitraps in the lab. We can do both of them, but one of them is not ideal. So in data-dependent acquisition, we already went through a little bit of this. What happens is that we are telling the mass spec, right, I want you to fragment always the 15 most abundant in each of the MS1. So 15 most abundant peptides that you see in one of the MS1, fragment them and then start and do an MS1 again. Then it isolates, fragments, and it goes back and it does this and until the end of the whole chromatographic run. Whereas in DIA, what happens is that it actually doesn't select anything. It does, but in much wider windows. So basically what we do is we tell the instrument, right, I want you to fragment everything at the same time, everything that falls between mass over charge ratio 1 and 43. That's an example, pure example. So it's going to select this window. So this is going to work still as a filter, but much more things are going through it, as long as its mass is between a window of M over Z. It sends off to the collision cell, fragments, analyze, and then you have a really complex spectra. But thankfully, nowadays, we have really good softwares to deal with this information. There's also more recent generation of mass specs that actually does this really fast. And by doing this really fast, what you can actually do is you can compress the window size that you're using in your DIA to the point that's actually exactly the same size of the isolation windows that you're using on your data-dependent analysis. So what's the problem here? Well, obviously, these mass specs are really, really expensive. They are really sensitive. They are really fast. You can actually have a full proteome coverage in 15 minutes instead of 3 days. For quantitative accuracy, you probably need to extend this time a little bit more, but they are really, really fast. So why are we doing DIA versus DDA? What's the story? So basically, it depends on the speed of acquisition of your mass spec. DDA is quite good, especially if you're having instruments like ours, which are Orbitrap instruments that are very accurate, but they are slow because that's what they do. It takes time for them to acquire. What happens is, because you're injecting your sample, one sample, then another sample, then another sample, because this is level 3, the order in which the most abundant peptides are selected is a bit stochastic. So it's not guaranteed that the same peptides will be selected from run to run, which means from sample to sample. And this creates missing values. In DIA, this is much more controlled because you're fragmenting literally everything that falls in the window. So your missing values is a slightly less of a problem. The only thing you need to, there's another example there. Okay. So, question for you. Which of these methods, DDA or DIA, allows you to do a full proteomic characterization? Yeah, well, good. That's exactly it. Both of them can. That's exactly what I was saying. So DDA, you can actually get a really complete proteomic characterization if you're using TMT, because TMT works with DDA. You actually have to get a really good isolation, as pure as possible. Not pure is not the correct term, but as specific as possible of the peptide that you want to fragment. Because if you have co-isolation of other peptides, when you're looking at the reporter ions, you're actually getting cross information about the quantification from different types of peptides. But because you can inject your fractions in 18 times, your sample goes in 18 times, each fraction has a different composition, you get full proteomic characterization. DIA also works like that, but especially with this new generation of instruments, the fast instruments, you can do it in much shorter gradient times. To get a good quantification, probably half an hour to 45 minutes per sample gets you around 9,000-10,000 proteins in one go instead of 3 days. And this is exactly what we get. So this is how the quantification is made. So on top, you can see, this is a chromatogram. You can see everything that went inside the mass spec. All those peaks are peptide signals, basically. And then what we do is we do extracted ion chromatograms, or not we, the softwares do extracted ion chromatograms, and then you can either use the intensity of that peptide, or you can use the area of the whole integration, basically, of the extracted ion. That's the signal for one specific peptide. In this case, you can actually see the mass here that has the signal for that specific peak. So you have basically one data. So what you're quantifying, even though you have MS2 data, you're quantifying at the MS1 level. Whereas in DIA, you actually have much more information that you can use towards quantification. You can use the extracted ion chromatograph. So again, it's the signal of the mass of the peptide, but you can also use the signal of the fragments of the peptide. So you have more information coming in. Now, I'm going to slightly change the topic. So this was a brief overview of the quantitative proteomics workflows that we have in the facility. Again, we also do these different things, and this is structural, going into the structural proteomics field. And the only difference between what I've just explained in this one is that we actually work with complexes or proteins that are in their native states. What we do is what we call cross-link them. And there's many different cross-linking agents that we can use. One that we use quite often in the facility is sulfo-STA, which has a moiety that will react with your peptides, where we incubate, it will attach to your peptides. And then it has a different group that will react with UV light. And we will create carbene groups that will highly reactive, that will either bind to the amino acids in proximity or to the backbone of the proteins, or amino acids in proximity in your protein. And then what we do is we digest. Certain amino acids would be glued, cross-linked to other amino acids, either from the same protein or from the neighboring proteins within your complex. And then what we do is we analyze by mass spec. Not really. So, because cross-linking efficiency is not as good as one would want for, what we have to do is we have to start with a really big amount of protein. We do the cross-linking assay, and then we digest and then we do a preparative fractionation by size exclusion, and we collect the fractions that obviously are bigger and they are more prone to contain cross-linked peptides because they are bigger in size. That's obvious. And then we run those in the mass spec. And then what we usually use is something that I haven't spoke, and I won't get into much detail, but it's a device that goes on the front of the ion source of the entry of the mass spec, and between the ion source and the mass spec, that mimics ion mobility. And basically, this is another way of selecting peptides. Depending on the voltage that you are applying, certain characteristics, physical properties of your peptides, namely the size, the shape, and the structure as well, we can select for those that are potentially cross-linked. So it's kind of a device that allows you to enrich in certain types of properties. and then it goes inside the mass spec. And in the mass spec, especially the one that we use is actually a very smart instrument in which we can create a hierarchy, not just say, right, fragment the most abundant ones. Now we actually want to look into different parameters, prioritize the bigger ones with more charges, because these are the ones that are going to be likely cross-linked peptides. And then fragment these, and then only look at the rest once you've done with this before. So that's how we do the cross-linking analysis. Another approach that we have in the lab, and hopefully in the future we'll update this one as well, is hydrogen-deuterium exchange. And this is very simple to understand. So you have your protein complex in its native state in an aqueous buffer. And then what this technique does is we plunge the protein complex in the deuterium, and the surface hydrogens will exchange very fast with deuterium, right? And then online on the mass spec, so this is not done on the bench, this is on the line. We have a pepsin column, which is a different protease that's going to digest these peptides. They're going to fly in the mass spec, and we're going to see the masses. So the peptides that have higher incorporation of deuterium will be the ones that are more exposed to it, whereas the ones that are more conservative in terms of the differences in masses are the ones that are protected from the exchange. So from this information, we can see which are the structures that are more superficial or have very variable regions, so we can study this. And now this is just the last slide to tell you that the next session, I'm going to speak a little bit about our bioinformatic pipeline, specifically one of them, which is kind of a new one in the LMB, and I want to help you to understand how you can use it to understand your data. And with this, I conclude my talk. Thank you so much. If you have any proteomics projects that you want to run, you don't know exactly what you need to do, just come on down. It will be our pleasure to show you actually around the mass spec room. It's a very noisy room with lots of instruments. We usually are very passionate about mass specs. So come in touch. Thank you.